And thank you very much for inviting me to come to speak. Um, let's think about phages in a very different way. So this is now 1 year out of date, and I apologize for not redoing it for this conference, but this is doing a basic search in the literature on publications in the peer-reviewed literature looking at phage. So this is really well done, guys. We've got a big dynamic field moving, but this is what has happened with prophage. And so the level of understanding of what prophages are doing to their bacterial host is really not as well studied as I think it can and should be. So lysogenic cycle, we all know. But here, with temperate phage, when you form the lysogen, you've got this huge piece of DNA, anywhere from 20 to, I don't know, 180 kb. And right now, the majority of people will look at this and they will look to see if they're toxin genes or their fitness genes or their antibiotic resistance genes. More frequently, they're not. And so if there's nothing interesting to see, it gets ignored. I started working with Shiga-toxin phages. And when I did this, I was a postdoc in the early days. And I knew that Shiga-toxin phages were lambda-like phages that encoded a toxin. And most importantly, they made E. coli 157 and then many other bacteria toxigenic. And so they were providing the primary virulence factor for E. coli to cause disease. This is an E. coli K12 strain and the subsequent Shiga-toxin phage. This was taken by Chloe James, who's in the audience, when she was a PhD student many years ago. So I knew that these were converting phages, and that's mostly what people think of when they think of prophages and their importance. So they encode something like this. So they have toxins or virulence genes, or maybe some moron, which is actually defined by a host gene that's been acquired and now carried on the phage. And that these genes directly have an impact on the host. And as we were going through this, we did some fairly standard types of experiments. And we looked this time, though, using a detoxified phage. Working with Shiga-toxin has to be done at Cat3. And I wasn't that keen on doing that for very long. So what we started looking at was identifying the receptor on the cell and how the phage recognized it. We looked at integration control because it was a little bit different than what you see with Lambda. We looked at induction controls because this was very different. C1 in the Shiga-toxin gene we were studying was absolutely a horrible repressor. It was very labile, it broke down very quickly. And so induction process was quite different. And we also looked at the burden of multiple prophage, STX prophage carriage, because most clinical strains of EHEC carry more than one Shiga-toxin phage. So did this actually have an impact on the amount of Shiga-toxin that was done? And so all of these questions were addressed and have been published. And we got sort of sidetracked by some interesting papers. And the first two were from Tim Wallace's group. And I remember right before these papers were published, he made a trip to Liverpool, and I had the opportunity to speak to him. And he said, in our STM mutagenesis studies, we found prophage gene mutants, but they're not Shiga-toxin. They're phage regulators. Do you have any idea why? I said, I have absolutely no idea, and put it on the back burner. Something similar happened with a virulent Pseudomonas aeruginosa strain, the Liverpool epidemic strain that causes really multiple drug-resistant infections in cystic fibrosis patients and is also transmissible from patient to patient, which is unusual for Pseudomonas. So the question started to become, what is the prophage doing? And this was our first attempt. So we threw our E. coli, lysogen and naive cells, on biolog, and we asked, do the lysogen and the naive cell behave differently? Now, we know that the prophage can be induced, so especially with some antibiotics, you might expect the lysogen to die off very quick, right? But this wasn't actually always the case. And sometimes the lysogen was really much more resistant to a perturbation in the naive cell. And we tried to make sense of this, but it was kind of hard to do. So first off, a prophage, as we know, is controlled by a repressor. So the genes should be turned off. So what was driving this? Well, we tried to answer this question with proteomic approaches, looking for proteins that were being expressed in the lysogen that weren't being expressed in the naive cell. This was almost impossible to understand. First off, we couldn't find repressor, so that didn't make a whole lot of sense. But secondly, we saw a lot of things like capsid proteins and tail proteins. So, well, those are late genes, so that doesn't make sense for a lysogen. So what we came up against was the fact that when you have a small population of cells in a lysogen culture undergoing spontaneous induction, which happens more times than I'd like to admit, that level of gene expression from the late genes is incredibly high, while the expression of prophage regulators in a stable lysogen is quite low. And so it's when you're looking for the end point, it's very difficult to tell them apart. So what we did was we moved on to RNA-seq. So this represents an induced lysogen, the naive cell also induced, treated the same way, the naive cell and the lysogen. And you might be able to immediately see where the prophage is in the hierarchical cluster. Now, what I want to point out to you is there are quite a few genes in the lysogen that are expressed that aren't part of induction. And we thought, well, it turned out there were a whole lot of host cell genes that were turned on, hundreds. And we looked at the level of phage gene expression. So here we have about a 58 kilobase genome. And these are the genes that we were found that were expressed from the prophage. And what we did to prove that they were prophage restricted is we did a qPCR profiling. And the way this works is if you take your stable cells and you induce them, and if it's prophage stable, after induction, you'll see it drop. And if it is induction sensitive, you'll see it quite low in the lysogen culture, but after induction, it rises. And you can also see that some genes are what I called uncoupled from phage regulation, and their expression doesn't change. So we saw things like LOM and BOR, which are found in lambda, were uncoupled from phage regulation, whereas we had many other genes, some of them regulatory proteins, that were expressed from the lysogen. Just picking the low-hanging fruit, because there were too many genes to look at, we identified that the E. coli lysogen had major changes in its motility-associated genes and in its acid adaptation genes. Now, it's already been shown that the GAD system was responsible for the real acidurants of E. coli 0157. And that's why those organisms are able to have such a low infection dose as they translate through the gut. And these are E. coli genes. So it was never questioned further. What we did was we discovered that these genes are actually upregulated significantly by a regulator on the prophage. And so these genes, though in the chromosome, are controlled by the prophage. And if you took an E. coli 157 strain and you remove that prophage gene, they were as sensitive to acid as K12. Moving on to a new pathogen, and I'm watching the time. Here we have our Liverpool epidemic strain of Pseudomonas aeruginosa. And when it was originally sequenced, there were 6 prophage areas that were identified. The first one is clearly not a prophage, but the others were, and Chloe James did this work again. She induced most of these phages from the cell, and we were able to put them into a related but different, less pathogenic background, into PaO1. And we began to study the impacts of these prophages. And because the other study hadn't been complicated enough, we took this one and made it ultra complicated. We grew our naive and lysogen cells under nutrient replete and deplete conditions. And we also looked at a whole lot of different lysogens. So only 3 of the phages in LesB58 can be integrated into PaO1 so far. So we used a combination of these 3 different prophages in multiple combinations as single, double, and triple lysogens, and compared them with induction and without induction through RNA-seq and these 2 different growth levels. And we looked to see what the changes were. Now, we had already very nicely seen that there were growth differences without seeing phage induction differences. And I still don't have a great way of showing this. This is looking at lysogen. This is looking at uninduced growth and induced growth in the naive cells under 2 different growth media. And then we have double lysogens, single lysogens, and triple lysogens, and the doubles. Okay, so there's loads of combinations there, but what I want to point out is, first, when you get an induction response, the level of gene expression goes through the roof. And when you're looking at lysogen gene expression, the levels are lower. But I also want to point out that they are all significantly different from the naive cell. And I think this picture does that well. What did we find? We found upregulation of gene efflux pumps. We found upregulation of alginate production. So far, we're talking about virulence factors encoded on the Pseudomonas. We saw differences in chemotaxis signal transmission. Now, we'd seen that in E. coli too. We saw lots of different metabolic pathway changes. And we saw biofilm-associated gene expression change. Now, we're in the process right now of mapping those phenotypes with all of the different lysogens under all of the different growth periods, and then testing those responses with particular prophase genes that cloned on an expression vector. So there's lots of work to do yet, but what it is clear is that there is an impact of gene expression on their host that has nothing to do with known virulence factors or anything else, because these phages don't encode those. And we also detect crosstalk in host phage gene expression between which phages are there. So phage 3 is good at downregulating some of the impacts of phage 2, for example. So Prophages are not silent passengers in their bacterial host. And prophages seem to, or are, able to adapt or evolve very quickly to be intimate partners with their bacterial host. Bioinformatic analysis may not really help predict what these genes are, because we saw genes that had these functions that were predicted to be regulators or involved in the phage life cycle and clearly have other roles. So prophages are going to be playing, and do play, because I can't believe it's just these two organisms, a fundamental role in the basic biology of their bacterial host. I think there's lots more to come, and it's sort of taking the time to unpick these things that will enable us to understand this better. So this is fundamental biology that changes our understanding of the bacterial host. And this has gone over many, many years. I started with a partner at the University of Liverpool, and I've had several. These are the people who have done, along with Darren Smith, because he did the biolog, who have done the shigatoxin phage expression work in Yue Yi, who has just become Dr. Yue Yi on Thursday. Yeah, she has a poster here to talk about motility impacts. And then these are people who have worked in the lab, both from the University of Liverpool and from Chloe James and Professor Ian Goodhead's lab, the University of Salford. And Lamia, who is just finishing her first year, has some information that she can share about the work that she's doing with Prophage 6. Thank you very much.